Cinnamaldehyde, that is the compound inside a $1 jar of cinnamon. It destroys fungal cell walls, not through one mechanism, through four, simultaneously. Peer-reviewed research confirms it inhibits the exact pathogens that cause root rot: Pythium, Phytophthora, Fusarium. The names that haunt every gardener who has ever pulled a plant from soggy soil and found brown mush where roots should be. Commercial fungicides cost $12 to $45 per bottle. They feed a global market worth nearly $24 billion a year. The alternative is sitting in your spice rack, right now, for roughly $1. But there's a catch nobody mentions. By the end of this video, you will know exactly what the science proves, what it does not, how to apply cinnamon 3 different ways, and why the one study that would settle this debate has never been funded. This is where we do things differently. The internet is drowning in miracle gardening hacks. Cinnamon cures root rot. Cinnamon replaces fungicide. Cinnamon does everything but pay your mortgage. And most of those videos never show you the actual research. They never tell you where the science stops and the assumptions start. This channel exists for people who want the real answer, not the easy answer, not the clickbait answer, the one you can actually trust when your plants are dying and you need to make a decision. If that sounds like you, subscribe. You are joining a community that demands evidence before action. We respect your intelligence here. We show the receipts, we flag the gaps, and we let you decide. So let us look at the receipts. Cinnamaldehyde makes up 85 to 95% of cinnamon essential oil. It is the dominant active compound, and it does not kill fungi the way most synthetic fungicides do. Most commercial products target one biological pathway, one lock, one key. Cinnamaldehyde targets 4. First, it disrupts fungal cell walls. The structural integrity of the cell itself is compromised. Think of it as dissolving the walls of a building while everyone is still inside. Second, it interferes with the tricarboxylic acid cycle. That is the central energy production pathway in fungal cells. Without it, the organism cannot generate the energy it needs to survive. The lights go out. Third, it inhibits protein metabolism. The cell cannot build the molecular machinery it needs to function. No new proteins, no repair, no reproduction. Fourth, it suppresses ATPase activity. ATPase is the enzyme responsible for energy transfer at the cellular level. Shut that down, and every other process grinds to a halt. 4 simultaneous attacks. This matters. When a fungicide hits one pathway, the organism can sometimes develop resistance. It finds a workaround. One door closes, the fungus finds a window. But when 4 pathways are disrupted at once, resistance becomes exponentially harder to develop. That is not folk wisdom. That is pharmacological logic. It is the same reason combination therapy works better than single drug treatment in medicine. Now, the numbers. Published peer-reviewed numbers. The minimum inhibitory concentration of cinnamaldehyde against tested fungal species is 60 microliters per liter. Against Phytophthora palmivora, one of the most destructive oomycete pathogens in horticulture, inhibition was observed at 1,000 microliters per liter. Complete inhibition of Aspergillus species was achieved at 60 micrograms per milliliter. And here is the detail that should get your attention. Cinnamaldehyde showed activity against drug-resistant fungal strains, strains that had already developed tolerance to conventional treatments. One more thing worth clarifying. Pythium and Phytophthora are technically oomycetes, not true fungi. They are often called water molds. This matters because some conventional fungicides designed for true fungi are less effective against oomycetes. Cinnamaldehyde does not seem to care about that distinction. Those numbers are real. They are published. They are peer-reviewed. And every single one of them was measured on a glass plate in a laboratory. Now, what you are hearing today is the surface layer of this investigation. Behind this video are hours of research synthesis, study cross-referencing, source verification. That deeper archive lives in the vault. It is where the full citations go. The extended application guides, the raw data breakdowns across dozens of topics like this one. No fluff, no filler, just the sources most channels never show you. If you want that level of depth, the link is in the description. But stay right here. You do not need the vault to use what I'm about to show you now. Here is where this gets complicated. Every single study I just cited was conducted in vitro, on a glass plate, in a controlled laboratory environment, no soil, no roots, no competing bacteria, no beneficial fungi that might be disrupted, no variable moisture levels, no fluctuating pH, no organic matter interacting with the compound, no sun degrading it, no rain washing it away. The leap from petri dish to potted plant has not been made in any published peer-reviewed study. The University of Saskatchewan stated this directly. Their position is that there is no scientific study to show that cinnamon works for damping off disease. And there is no recommendation about the amount to use. That is not a fringe opinion. That is an institutional statement from a major agricultural research university. And there is another problem most cinnamon videos never mention. The studies used concentrated essential oil or purified cinnamaldehyde, isolated, refined, measured in precise microliter and microgram quantities. What gardeners use is grocery store ground cinnamon. The concentrations are orders of magnitude apart. It is the difference between medical-grade aspirin and chewing on willow bark. The active compound is present in both, but the dose is not remotely comparable. So if I stopped here, I would be telling you cinnamon does not work, but that is not what the evidence says either. The evidence says we do not know. The lab science is strong. The field science does not exist. Those are two very different statements. Absence of evidence is not evidence of absence. So why has nobody run the study that would settle this? Follow the money. The global fungicide market is valued between $19.6 billion and $23.9 billion annually. That is billion with a B. The products lining garden center shelves cost $12 to $45 per bottle. They are designed to be reapplied season after season. That is a recurring revenue model. Shareholders love recurring revenue. Now, consider cinnamon. You cannot patent it. It is a commodity agricultural product. Cinnamaldehyde is a naturally occurring compound. There is no proprietary formulation to license, no branded molecule to protect, no subscription model, no recurring purchase cycle. A gardener buys one jar for $1 and potentially uses it for years. There is zero commercial incentive for any company to fund the field trial that would validate cinnamon as a fungicide alternative. Zero. Not $1 of profit to be made. Not one quarterly earnings call that gets better because someone proved a spice rack staple works on root rot. And this is not conspiracy. I want to be precise about that. I am not telling you that a shadowy boardroom decided to suppress cinnamon research. What I am telling you is something simpler and more defensible. The system is not designed to validate cheap solutions. The pharmaceutical industry works the same way. No company funds large-scale trials on unpatentable generic compounds, not because they are afraid the compounds work, because even if they do work, there is no return on investment. The absence of proof becomes, functionally, market protection. Nobody had to ban anything. Nobody had to suppress anything. The research just never gets funded. And without funded research, there is no official recommendation. And without an official recommendation, the institutional answer remains what the University of Saskatchewan said. We do not know. Use the commercial product. I am connecting documented facts here. The market size is documented. The lack of patentability is documented. The absence of field trials is documented. The connection between those facts is my framing. Reasonable people can disagree about whether that framing is fair. I think it is. But I want you to see the seams, but none of that changes what you can do in your own garden. Today, for $1. If this kind of investigation matters to you, if you want a channel that separates what is proven from what is marketed, hit subscribe. That is how you stay connected to this work. Not hype, not conspiracy, just honest investigation. Now let me show you exactly how to use this. But first, ground rules. Cinnamon is preventive. It is not curative. If your plant already has established root rot, cinnamon will not reverse the damage. The infection is systemic at that point. The tissue is dead. You need to cut away affected roots, improve drainage, and possibly apply a targeted treatment. Cinnamon does not contain auxins. It is not a rooting hormone. Some sources online claim it stimulates root growth. That is not supported by the science. It may protect a cutting from fungal infection during the vulnerable rooting phase. That is different from stimulating root development. Do not confuse those two things. Thick application causes problems. A heavy layer of cinnamon powder on soil becomes hydrophobic. It crusts. It repels water instead of letting it reach the roots. More is not better. Thin and even is the principle. And the most important thing I will say in this entire video. The number one defense against root rot is, and always has been, proper drainage and responsible watering. Period. Cinnamon is a supplement. It is not a replacement. If your pot has no drainage hole, no amount of cinnamon will save your plant. Fix the fundamentals first. With those ground rules clear, here is how you actually do it. Method 1. Soil surface dusting. Take ground cinnamon and apply a thin, even layer across the top of your potting soil. Emphasis on thin. You want a light dusting, not a crust. A quarter teaspoon spread across a 6-inch pot is sufficient. After watering, some of the cinnamon will wash into the top layer of soil. That is fine. Reapply after each watering if you want to maintain the barrier. This method creates a surface-level antifungal environment. It does not penetrate deep into the root zone. It is best suited for preventing surface mold and damping off in seedlings. Remember, the concentration reaching your plant roots is far below the minimum inhibitory concentrations measured in lab studies. You are working with grocery store powder, not purified cinnamaldehyde. Method 2. Cinnamon water drench. Add 2 to 3 tablespoons of ground cinnamon to 1 quart of warm water, not boiling, warm. Steep for 8 to 12 hours. Overnight works. Strain through a fine mesh sieve or cheesecloth. You want the liquid, not the sediment. Apply as a soil drench, pouring directly onto the potting medium. You can also use a spray bottle for surface application on stems and lower leaves where fungal issues tend to appear. This delivers cinnamaldehyde in solution, which penetrates more effectively than dry powder. But I must be honest with you. Even this concentration is still far below the MIC values published in the research. You are making an informed bet, not following a proven protocol. Method 3. Propagation wound treatment. When you take a cutting from a plant, the fresh cut end is an open wound. It is vulnerable to fungal colonization before callousing occurs. Dip the fresh cut end directly into dry cinnamon powder. Tap off the excess. Then, place the cutting into your rooting medium. This creates a localized antifungal barrier at the most vulnerable point during the most vulnerable window. It does not replace rooting hormone. If you want to use rooting hormone, apply it separately. Cinnamon is providing pathogen protection, not growth stimulation. Preventive only. 3 methods, $1. And a question that still does not have an official answer. Here is where we land. Cinnamaldehyde is a real compound with real, peer-reviewed antifungal properties. 4 mechanisms of action. Activity against the specific pathogens that cause root rot. Activity against drug-resistant strains. FDA generally recognized as safe status, which means it will not harm you or your soil biology in normal use, and a price point of roughly $1. All of that is verified. What is not verified is whether ground cinnamon, at the concentrations a home gardener can achieve, delivers enough cinnamaldehyde to meaningfully suppress soil pathogens in real growing conditions. That study has not been done. The reason it has not been done is structural, not conspiratorial. Nobody profits from proving a $1 spice replaces a $45 product in a $24 billion market. So you have a choice. An informed one. Cinnamon is safe, it is cheap, it is lab-backed, and it is worth trying as a preventive supplement alongside the fundamentals that actually matter. Good drainage, appropriate pot sizing, responsible watering. Those are your real weapons against root rot. Cinnamon is a low-cost, low-risk addition to that arsenal, not a replacement for it. Experiment. Observe your own results. Compare treated pots to untreated ones. Be your own field study. That is more than most gardeners ever do. And if someone you know is about to spend $45 on a bottle of fungicide for their houseplants, maybe send them this first. Let them decide with the full picture. Next week, We're going into the vault on a compound that the medical industry quietly removed from over-the-counter access in 1971. The timing lines up with a patent filing that nobody talks about. You are going to want to see that one.